Okay, so to kick things off, we're going to start with our trainee speaker. We have postdoc Dr. Charneal Dixon from Dalhousie University, who's going to tell us about inhibiting ABHD17 proteins and how that restores S-palmitoylation and membrane localization. So Charneal, you can take it away.
Okay, let me just share my screen. Okay, is that good?
Yeah, looks great.
Okay, so hello everyone. Very quickly, I just want to thank our host CSMB for this excellent seminar series and for creating a platform for trainees like myself to be able to share our research. So the talk that I'm giving today is going to focus on the nucleotide binding oligomerization domain containing protein NOD2 and the recent progress that we've made in identifying NOD2 depalmitylating enzyme and the importance of those enzymes in regulating NOD function. So a bit of background just to kind of give some context for this project. Crohn's disease is a type of inflammatory bowel disease that can affect any segment of the gastrointestinal tract, extending from the mouth all the way to the anus. However, it does most commonly manifest itself in the lower portion of the small intestines and the upper colon. In Nova Scotia, my home province, Crohn's disease is a growing concern, with 1 in every 80 people affected by this condition. And the burden of IBD in Canada does appear to be rising. In 2018, there were roughly 270,000 Canadians living with IBD. And last year, those estimates were up by more than 50,000. Now, since the NOD2 gene was first characterized in the early 2000s, it has been consistently implicated in the pathogenesis of Crohn's disease. So on the right, I have a little schematic here. NOD2 is a cytoplasmic microbial sensor that surveys for fragments of bacterial peptidoglycan after it's been transported across the plasma membrane. It triggers pro-inflammatory responses that are mediated by NF-kappa B and MAP kinase signaling pathways. And all of these responses are essential to the bacterial killing activity of Paneth cells via their secretion of microbiocidal compounds. Now, it's long been known that about 20 years that NOD associates with the plasma membrane, but it's only in recent years that we've determined that protein palmitylation is the mechanism for this association. So the protein acyltransferase enzyme ZDH8C5 mediates the addition of fatty acyl groups to NOD2, which increases its hydrophobicity and its affinity for membrane-bound, for the plasma membrane or membrane-bound compartments like endosomes. This is absolutely critical to NOD2's ability to respond to an immune insult and promote clearance of bacteria. Now, so the work that we're doing that I'm presenting today is actually going to be building on all of that research. We've identified the thioesterase that's responsible for removing those palmitates and allowing NOD2 to re-enter into the palmitylation cycle. Unlike other types of protein lipidation, the covalent attachment of fatty acids to cysteine residues via a thioester linkage, it can be reversed. Now, I'll say that prior to this study, nothing was known about the reversibility of this modification on NOD2, and there were absolutely no clues as to the identity of the thioesterase that was depalmitylating NOD2. So we used a colorectal epithelial cell line and developed a cell line that had controllable low-level expression of GFP NOD2. Now, the first thing we had to establish with these cells was that they actually contained an enzyme capable of depalmitylating NOD2 so that we could illustrate that protein palmitylation was reversible and it was enzymatically mediated. So as you can see from the DMSO control here in these cells, when NOD2 is expressed, it's a very large cytoplasmic pool with only a very small portion of the protein actually associated with the plasma membrane. Now, when we treat cells with palmistatin B, which is a cell-permeable, broad-spectrum inhibitor that's known to target multiple depalmitylase enzymes, we saw a significant increase in the fraction of NOD2 that was associated with the plasma membrane. Now, since it was previously demonstrated that NOD2 mutants that are incapable of being palmitoylated are completely cytosolic, we reasoned that this observed increase in the plasmalemal enrichment of NOD2 was likely going to be due to a more significant fraction of the protein also being palmitoylated. So to confirm this experimentally, we used a method called acyl resin-assisted capture. It's a method of isolating palmitoylated proteins, and it has a series of steps, beginning with first blocking any free thiols, and then cleaving off any acyl thioester bonds, so any palmitate or fatty acyl groups that are attached through a thioester linkage, using neutral hydroxylamine, and then using an affinity-based resin or a thiol-reactive resin to then capture that protein based on the newly liberated thiols. Now, what we found when we immunoblotted for GFP NOD2 after doing acyl-RAC was that we were able to capture almost, well, more than double the amount of palmitoylated protein in cell lysates from cells that were treated with palmistatin B compared to the DMSO control. So when we combine the confocal images that I showed you and the results of our acyl resin-assisted capture, it suggests that protein palmitylation of NOD2 is reversible, and that these cells contain a protein that is sensitive to palmistatin B that's mediating the enzymatic removal of palmitate from NOD2. So encouraged by our work with the broad-spectrum palmistatin B inhibitor, we then curated a selection of small molecule inhibitors that are known, well, at the time, we got inhibitors that were known to target all the known depalmitylation enzymes. There have been new discoveries of depalmitylation enzymes that are not shown here. So in the table on the left, what I've shown were the chemical inhibitors that we were using and their targets. But on the right, I've only shown the confocal images for those inhibitors that were effective in changing the localization of GFP NOD2, namely the lipase inhibitor THL and the selective ABHD17 inhibitor ABD957. And as you can see from the quantification graph below, there was almost a 5-fold increase in NOD2 enrichment on the plasma membrane when ABHD17 enzymes were chemically inhibited. So again, we were able to confirm using acyl resin-assisted capture that the observed increase that we saw in the plasma membrane corresponded to increased palmitylation. Now, while we did observe a modest increase in the plasma membrane localization when we were using the THL inhibitor, there was no significant increase in the amount of captured palmitoylated GFP NOD2. However, when we were using the ABD957, the ABHD17 inhibitor, we saw a 4-fold increase. Okay, so having narrowed down a possible list of candidate enzymes using chemical inhibitors, we next examined the cellular consequences of overexpressing the individual thioesterases that were targeted by ABD957. So we knew from prior work that preventing NOD2 palmitylation, either by mutating cysteines or treating the cells with chemical inhibitors like 2-bromopalmitate, it would prevent NOD2 from localizing on the plasma membrane. So we expected overexpressing the enzyme that's capable of depalmitylating NOD2 to similarly reduce the fraction of GFP NOD2 that we would see on the membrane, if that makes sense. So we found that either of the 3 ABHD17 isoforms was able to accomplish this, with 17B and 17C appearing to be the most effective. And I've highlighted the cells with an asterisk, the ones that were co-transfected with both the thioesterase and the GFP NOD2. So for us, the next logical step was to determine if the endogenous ABHD17 enzymes regulated NOD2 depalmitylation in these cells. So we were essentially just investigating the effects of using RNA interference to knock down the ABHD17s and then looking at NOD2 palmitylation. Now, when we silenced the 17A isoform, there was no significant change in the localization of NOD2. However, there was a modest change for 17B and 17C. The simultaneous knockdown of all three of the isoforms, however, resulted in an almost fourfold increase. And furthermore, the triple knockdown results were comparable to what we saw when we were using the selective ABHD17 inhibitor. So collectively, the results from both our overexpression experiments and the silencing experiments were indicating that the ABHD17B and 17C are likely the primary thioesterases that are responsible for NOD2 depalmitylation in these cells. So we also examined how inhibiting or silencing ABHD17 would affect endogenous NOD2 activation and downstream pathways. So to do this, we monitored the phosphorylation status of downstream effector proteins for NF-kappa B. So we used P65 and IKB alpha, and then P38 for the MAP kinase signaling pathway in response to NOD activation. So these cells were stimulated with MDP. Now, based on previous studies, we again knew that inhibiting S-palmitoylation of NOD2 would reduce NF-kappa B and MAP kinase activity. We observed this both in raw cells and in the same colorectal cells that we're using here for this study when the cells were treated with MDP. So we are fully expecting that the increased palmitylation from attenuating ABHD17 would result in an increase in NF-kappa B and MAP kinase responses. And as predicted, cells that were chemically inhibited with the ABD957 and those that were depleted of ABHD17 using siRNA interference displayed an increase in both NF-kappa B and MAP kinase responses. We also took it a step further. Since both NF-kappa B and MAP kinase activation has the capacity to induce transcription of pro-inflammatory cytokines, we wanted to determine if the enhanced signal transduction that we saw would result in an increase in cytokine production. So both the chemical inhibition and the depletion of ABHD17 increased inflammatory cytokines, most notably GM-CSF and IL-8. So we see that the loss of the ABHD17 function increases the level of palmitoylated NOD2, which subsequently increases the activity of NF-kappa B and MAP kinase signaling pathways that regulate the production of pro-inflammatory cytokines. That's a mouthful. Finally, we had also previously reported that several Crohn's disease NOD2 loss of function variants have reduced palmitylation levels. Naturally, we wanted to know if targeted inhibition of ABHD17s could potentially rescue these mutants. And by rescue, I mean restore the mislocalized protein to the plasma membrane with a corresponding increase in palmitylation, as well as improve signal transduction in response to MDP. Now, we weren't able to rescue every mutant we tested, including the leucine 1007 frameshift mutation that I'm showing right now. This mutation results in a truncated protein that is absent one of the two mandatory palmitylation sites. So we couldn't rescue this mutant by inhibiting ABHD17, but we were able to rescue some of them. And just for the interest of time, I've isolated one example. Using confocal microscopy, we saw that the A755 valine Crohn's disease mutant, which is a completely soluble protein when expressed in cells, started showing relocalization to the plasma membrane We also saw an increase in the amount of palmitoylated protein that we were able to capture in the lower left-hand corner. And we saw an increase in signal transduction when we again looked at downstream phosphorylation of our downstream effectors. And then lastly, we saw an increase in the pro-inflammatory cytokine secretion, mainly the IL-8. So now we have a more complete picture of NOD palmitoylation, both chemical inhibition and siRNA-mediated depletion of ABHD17 enhanced the plasma membrane localization of NOD2, which corresponded to an increase in S-palmitoylation of the protein, as well as an increase in MDP-mediated signaling and cytokine expression. And as I've hopefully illustrated, despite only showing one example, we know that this targeted inhibition of ABHD17 could prove to be advantageous for even a subset of these Crohn's mutants. It at least could be beneficial therapeutically. And with that, I'm just going to leave up my acknowledgement slides because I believe I'm at 15 minutes. So I won't have time to mention every single person, but I will say that this work was made possible through the contributions of a number of people, a number of great thinkers. So for anyone that's interested in gaining additional information on the work that I've presented here, a preprint is available and the QR code is located in the bottom right-hand corner. And of course, I am happy to try to answer any of your questions.
Awesome. That was a great talk. Thank you, Charneal.
That was fast.
Yeah, for sure. But you did a nice job of illustrating your work, I think. So any questions now? Like I mentioned at the beginning, feel free to either raise your hand and we can unmute you, or you can type them in the chat. I can maybe start with a question while people are thinking of some of theirs. So are there any known variants in the ABHD that are associated with Crohn's disease or other inflammatory diseases?
That is a great question. And no, we don't know of any that are associated with Crohn's disease.
Okay.
The interesting thing to note is, I think it was somewhere in the last 10 years that the ABHD17 family was really identified. So not a whole lot is known. The inhibitor that we were even using was created by our collaborator in the midst of this. So we were actively reading papers and finding out about new thioesterases and new inhibitors as we were developing the project.
Okay, very cool.
That's a great thing for someone to look into.
Right, cool. Dr. Mitchell?
Yeah, really great talk. I guess somewhat on a similar vein, I was wondering, do you know if any of the current treatments for Crohn's disease may actually be affecting this process and it was just sort of not known before, or?
No, I don't know, but it's interesting. Someone mentioned to me when I gave a talk the other day that TNF alpha was one of the primary biologics, like anti-TNF alpha medication, which I thought was interesting. I actually want to go back and dig through my list of cytokines to see if I was seeing a change for that one, maybe a decrease. But no, I don't know of any offhand.
Great. Any other questions from anybody else in the audience? Okay, seeing none, let's thank Charneal one more time.
Thank you.
And we'll move on
I'll stop my share.
Perfect. Okay. I think this is going to pull up in a minute. There we go. So we'll move on to our next talk. So next we have Dr. Jennifer Mitchell from the University of Toronto, who will tell us about finding function in the non-coding genome. You can take it away, Dr. Mitchell.
All right. Okay, do you see my full screen?
Yeah, your slides look good.
Okay, and hopefully they will advance properly. Okay. Whoops. Let me just get a laser pointer, which will help. All right. So I'm going to share some of our work looking for gene regulatory elements in the non-coding genome and trying to understand how they function. Okay. So if you think, about the size of the human genome, it's really massive. It's vast. So it's around 6 billion letters, and that can be sort of hard to visualize. And so I sort of looked for correlates, and the estimated size of English Wikipedia is close to 6 billion words. So it's sort of a good correlate. And there was this image put out a while ago to sort of say, well, what would it look like if we printed all that out and put it in old school kind of encyclopedia volumes? And so here that's pictured with a person, typical height of a person standing next to it. And so we're talking about this massive, massive amount of information. But actually, at a sequence level, we really only understand the meaning of less than 2% of the sequence. And by that, I'm referring to the coding regions. And just to map that back onto our sort of analogy for the size of the genome, that's what that looks like. And so you could say, okay, well, if we have a decent understanding of how to interpret sequence in the coding regions, isn't function all about the genes? And that's all we really need to know. And in fact, that's not the case. Of course, genes are really important, but equally important in understanding disease and evolution and the functioning of our genome is understanding the regulatory sequences that control the expression of genes in particular time and place and in response to specific signals. And so in mammalian genomes, transcriptional regulation is very complex. There are many different types of regulatory elements. I'm going to be mainly focusing on one type called an enhancer. So enhancers will enhance the expression of their target gene. And unlike promoters, which are located quite close to the transcription start site of the gene, enhancers can be close or they can be millions of base pairs away from the gene that they regulate. And that presents a lot of problems in terms of figuring out where they are and exactly how they function. We know a fair amount about their chromatin biology and context. So enhancers tend to be more nucleosome-free regions, meaning that they're more accessible DNA compared to the regions that are tightly packed with nucleosomes. They are bound by transcription factors, which are DNA binding proteins that bind to specific short sequence motifs in the DNA and can also cause increased chromatin accessibility at the regions that they bind. The picture is even more complex because these transcription factors can recruit other proteins called coactivators. I'm just showing you a couple of examples here. So there's one coactivator called EP300, which is actually an acetyltransferase enzyme that will acetylate histones close to the regions that it's associated with. And so this enzyme will actually put a lysine 27 acetylation onto histone H3. It also acetylates a number of other proteins, but the histone acetylation is sort of most well studied in terms of enhancers. Enhancers are also associated with other histone modifications. The sort of second most prevalent one is this histone H3 lysine 4 monomethylation that's often found at active enhancers. The transcription factors will also recruit a large complex of proteins called the mediator complex. This is a complex of more than 30 proteins, which is involved in interacting with the basal polymerase machinery that is associating with the promoter of target genes. And in fact, part of the function of enhancers, the way that they can actually function at a distance, is that they're known to form these chromatin loops whereby the enhancer is brought into close proximity to the gene promoter that it's regulating. And that's one of the ways that we can try to find these enhancer promoter regulatory relationships. I mentioned previously that enhancers are actually really important in regulating tissue-specific expression. They're actually thought to be the main mediators of tissue-specific expression. Rather than the gene promoter that might cause low-level expression in a wide variety of contexts. So in contrast to that, enhancers can be highly tissue specific. What I'm showing you here is a strong enhancer in the forebrain, and this is one of the currently validated enhancers from the VISTA enhancer database, which has identified the tissue specificity for around 2,000 enhancers, about half of which are human sequence and half are mouse sequence. And these, I wouldn't be talking to you today if these enhancers didn't have a really important role in cell homeostasis, development, and also causing disease. And we know this because deletion or mutation of enhancers can cause diseases and also lead to developmental phenotypes. And we know that this is actually quite widespread from a large number of genome-wide association studies. So what's a genome-wide association study, or GWAS? So this is an experiment that allows us to identify potential genetic links to disease. And if we do this for human patients compared to controls, then we can learn about genetic links to human diseases. So how this is done is that a number of controls are identified, and a number of patients for a particular condition or phenotype. In this case, I'm showing you an MS related study. And the genotype of these individuals is gathered either using a single nucleotide polymorphism array, or more recently using whole genome sequencing. And so this then allows us to make a link between a single nucleotide polymorphism in the patient that is more associated with disease. And there's in fact a huge database of these GWAS links, which is now around just over 600,000 unique single nucleotide polymorphism trait associations from close to 7,000 publications. And these are widely distributed across the human genome. So these are cartoons of the individual chromosomes and regions where these links have been identified. And now if we sort of zoom in closer and try to say, okay, well, where are these links? Are they more prevalent in genes, or are they not? And in fact, early studies identified that the GWAS SNPs were actually not that enriched in genes. So less than 5% of them are found in coding regions, and more than 90% are found in the intergenic regions of the genome, with many of them predicted to be actually affecting enhancer type regulatory elements. And so this is telling us now that from these 600,000 disease associations, a very large number of them are likely to function through modulating the gene regulatory sequences rather than having changes associated with the gene itself. So here's an example from a very well-studied locus. This is the beta globin locus, and this is actually a cluster of the beta globin genes that are expressed at different times during development. And this was actually the first identified distal enhancer that drives the expression of a mammalian gene. And it was identified because enhancer deletions were found in patients with a rare blood disorder called beta thalassemia, which is due to low expression of beta globin genes. And it was determined that these patients didn't have anything wrong with any of the beta globin genes, but they had this deletion of a region upstream. And it was later determined that this region actually acts as an enhancer to enhance the expression of the beta globin genes in erythroid cells that will later become mature red blood cells. And so this enhancer has been studied for more than 50 years, but we still don't have a complete picture of how the sequences within this enhancer are functioning. And I just wanted to highlight here that there's a GWAS SNP that's found within this enhancer that is linked to hemoglobin concentration in the blood. And so this is not a disease state, but more of a phenotype that individuals with a different version of this SNP will tend to have a different concentration of hemoglobin compared to individuals with the other version of the SNP. And we actually still don't really understand how that polymorphism is actually affecting the function of this enhancer. So there's still a lot to be learned, even for enhancers that we've been studying for many, many years. Now, my lab doesn't work in this blood system, but my lab actually works mostly with stem cell models. And we started off with pluripotent stem cells, which are a really great system for studying transcriptional regulation for a few reasons. So one is that early on, we had a really good idea about some of the critical transcription factors that are important for maintaining this pluripotent state. And that is because Takahashi and Yamanaka had shown that expressing these 4 transcription factors in a fibroblast would drive the cell to this induced pluripotent state, indicating that these transcription factors are really driving the acquisition of a pluripotent state, meaning that they're really important for the transcriptional regulatory network. And also around the same time, a number of chromatin immunoprecipitation sequencing experiments started to determine where different transcription factors were binding the genome on a genome-wide scale in these cells. So there was a lot of data for us to use to try to formulate hypotheses about where the enhancers might be and how they might be regulating specific genes. And we initially asked the question, how is the SOX2 transcription factor, how does it become highly expressed in the pluripotent stem cell state? And our sort of bread and butter experiment to really identify where a critical enhancer is became a CRISPR deletion. So we would use guide RNAs to target regions bound by transcription factors and delete a large chunk of the genome to remove a presumptive enhancer, and then ask the question, what happens to gene expression? We also have another trick that we use in these stem cell models, because we use cells that were generated by crossing 2 different strains of mice that have a lot of polymorphisms between them. And that means that we can independently look at each allele, and we know the allele that's the maternal origin allele and the paternal origin allele. So we can make a deletion on only one of those 2 alleles and look at how it affects the expression of the gene on the linked allele. And so here I'm showing you a representation of the region of the genome that contains SOX2. Here's the SOX2 gene. It's a tiny, small intronless gene. And surrounding that gene, we found a number of regions that were bound by transcription factors. So that's all of this data down here that summarizes the chromatin immunoprecipitation sequencing data that we had. And we became really interested in this region. And we showed, using a number of approaches, that there were 2 classical enhancers in this region. And so this was actually our first CRISPR deletion. We used guides to take out this entire chunk of DNA. And then we just did genome-wide RNA sequencing to look at any changes in gene expression. And when we only deleted this region on one allele, we found that SOX2 was reduced in expression on that same allele. And we then called this region the SCR for the SOX2 control region, knowing now that this region was actually regulating transcription of the SOX2 gene. And so in these cells where we've only deleted one of the enhancers, we actually didn't really see the cell state change at all, because expression of SOX2 from the other allele was enough to support the pluripotent state. But now what happens if we actually delete the enhancer on both alleles? So when we and then look at genome-wide sequencing again. So when we delete the enhancer on both alleles, we still see a reduction in SOX2 expression, but now we see more changes globally to the transcriptome. And very strikingly, we see this upregulation of a number of genes that are not supposed to be expressed in ES cells, indicating that these cells are kind of losing their cellular phenotype. We also saw a downregulation of a number of genes on the X chromosome, and these cells are actually female, and in the undifferentiated state, they have 2 active X chromosomes. And so this downregulation of X-linked genes is likely due to the fact that the cells are undergoing X inactivation, which is a measure of the cells starting to differentiate. And so deletion of the enhancer on both alleles actually causes the cells to become partially differentiated, telling us that we really need this enhancer to maintain the undifferentiated pluripotent state. But now you might have noticed that there's something interesting going on closer to the gene. And after having looked at this very far away enhancer, which is over 100,000 bases away from the gene, we were kind of curious to see, well, what about these regions closer to the gene? Are they important? Are they enhancers that you maybe need these to function with the distal one? And we did a number of deletions around here that I'm not going to show you because they're published, and we actually saw no difference in SOX2 expression. And so we were a little bit disappointed by doing a lot of work and seeing nothing, but we thought, well, what if these regions closer to the gene are actually important in another context? And so we sort of turned to thinking about what happens later in development. So we know that SOX2 is really important in the pre-implantation blastocysts in what's called the inner cell mass. This is the pluripotent population of cells at this time. And if you delete the SOX2 gene on both alleles, the embryo dies. The embryo cannot continue to develop beyond implantation. And so this is peri-implantation lethal. Recently, collaborators of ours at the NIH deleted the SCR in this context and also showed that that had the same phenotype as the gene deletion. So that was nice. But a lot of earlier work had shown that SOX2 is also expressed in developing nervous tissue. And so we thought maybe those enhancers closer to the gene might be important at that stage. And from a human perspective, it was interesting to also consider this because SOX2 haploinsufficiency, so mutation of just one of the genes in the human population, causes a disruption to eye development as well as brain development. And so it did seem like SOX2 was probably going to be quite important in this context. And so we made use of the differentiation potential of embryonic stem cells, and we then differentiated them into neural stem cells and asked about their regulation in this context. And this was work that was done by two postdocs that have sadly now left my lab. One of them is a new professor at the University of Guelph, Ian Tobias, and Sakti Murthy is now working with the Center for Commercialization and Regenerative Medicine, where they work to support regenerative medicine translational work by the research community in Canada. And they've done a lot of experiments that I'm not going to show, but just briefly, what they found out is that if you take cells that have an SCR deleted and you look at their expression in the embryonic stem cell state, you see that their expression is very low compared to wild type, shown in blue. But now if we differentiate those cells to neural stem cells, they actually recover their SOX2 expression just fine. So this suggested that there were probably other enhancers that were driving SOX2 expression in this context. And in fact, they seem to be these regions closer to the gene, since if you delete those regions, you now don't see an effect in embryonic stem cells. They look very similar to wild type in terms of their expression levels. But if you differentiate them to neural stem cells, you now see a disruption in SOX2 expression in that context. And so those proximal enhancers are required for SOX2 expression in neural stem cells. And in fact, if we look at the histone modification that I told you is associated with enhancers, we see that that's much stronger in this region when we're looking at brain tissue compared to embryonic stem cells. And so we now know that actually these enhancers are important in maintaining a more anterior regional identity of these neural stem cells compared to the more posterior identity that we see when we delete these enhancers. But I'm not going to get into more detail about that now. And so just in summary for what I've shown you so far, there are these neural lineage enhancers that are close to the SOX2 gene. 100,000 base pairs downstream of the SOX2 gene are these embryonic stem cell enhancers that were important in embryonic cells. And we were next interested in looking at what about contexts like cancer. So how does SOX2 expression become misregulated in cancer? And our initial hypothesis was maybe that these embryonic stem cell enhancers might be getting reactivated in a cancer context. And so SOX2 is really overexpressed in a number of different cancers, where in most of these cases, it does seem to drive a more tumorigenic and more disruptive phenotype. And one of the reasons why we were particularly interested in looking at how SOX2 is regulated is because SOX2 is one of what's called a pioneer transcription factor, where it can scan very closed and inaccessible chromatin that's very tightly associated with nucleosomes. It can find the DNA binding motif that it usually associates with and recruit other modifying enzymes that can then increase the accessibility of that chromatin. And so SOX2 is then able to transform in this manner both the epigenome and the transcriptome, and that can then have widespread knock-on effects for the cellular phenotype in cancer cells. And so we were interested to try to figure out how SOX2 is getting misregulated in these different types of cancer. This was largely the work of a fantastic PhD student, Luis Abati, who graduated from my lab a year and a half to 2 years ago. And his work was published fairly recently in the journal Nucleic Acids Research. And so what he saw was that SOX2 was overexpressed in a number of different cancer subtypes, but breast and lung were particularly interesting since they showed a really large increase compared to normal tissue. If he looked in the database that has both patient outcomes and RNA sequencing, he found that the patient tumors that have low SOX2 expression tend to do better. Survival is better compared to the group that has high SOX2 expression where survival is worse. And so it does seem like SOX2 expression is actually affecting patient outcome in a number of different tumor contexts. And so we wondered, if we look around the SOX2 gene, can we find any regions that might be enhancing expression in tumors? And I've told you a few times that enhancers are more accessible open DNA. And there's this assay called Assay for Transposase Accessible Chromatin, or ATAC-seq, that you can use to identify these more accessible chromatin regions. And it's a genome-wide assay. And there is a large database of patient tumor samples that have been subjected to ATAC-seq, and most of these samples have a matched RNA-seq data, so we can compare chromatin accessibility and gene expression. And so I'm just showing you a snapshot here of a few different patient samples across a number of different tumor subtypes. And I told you, here's our SOX2 gene over here, again, our small intronless gene, those neural enhancers closer to the gene. And then here's the human equivalent of that pluripotent embryonic stem cell enhancer, which we thought might be becoming reactivated in different cancer contexts, but actually we haven't really seen any good evidence for that. Instead, Luis found these enhancers that are a little bit closer to the gene, but still quite far away, that are 124 and 134 kilobases downstream of the human gene. And these seem to be becoming accessible in a large number of different subtypes of cancers. And this was a little bit unusual because we were, as I told you before, enhancers are generally very tissue specific. So we weren't really expecting to see that a number of different cancers might have the same enhancers becoming activated. And so we wanted to look at these in more detail. And then next thing that we did is look at how the accessibility at these regions is correlated with SOX2 expression. And so some patient tumors have more accessible chromatin at these regions and some have less. So we split the samples into the ones that had low accessibility or the ones that had high accessibility. And then we looked at SOX2 expression in the RNA-seq data. And we saw that for both of these regions, the highly accessible the patient tumors that had more highly accessible chromatin had significantly higher SOX2 levels, suggesting that this region could be regulating SOX2 in these tumors. But then, of course, we wanted to turn to some more functional assays. And so we needed some cell line models. And so for breast cancer, we turned to MCF7s and T47Ds. And we saw that in both of these cell lines, they had more accessibility at these two regions compared to the breast tissue where we didn't see, to normal breast tissue where we didn't see accessibility. They also had those histone modifications associated with enhancers in both contexts and evidence of a chromatin loop that could link the 134 region to the gene promoter, which is shown in pink there. In lung cell lines that we looked at, we also saw increased accessibility at these regions and histone modifications compared to the normal lung tissue. And so from this, we picked out some cell lines that we could work with further. And now we wanted to first do the CRISPR deletion to see if these regions really are required for SOX2 expression in cancer cells. And in both MCF7s and PC9 breast and lung cells, we saw this massive reduction of SOX2 expression down to almost undetectable levels when the enhancer was deleted on both alleles, indicating that yes, this region is absolutely required for SOX2 expression in these cellular contexts. And we also saw a reduced clonal growth in an in vitro clonal growth assay when the enhancers were deleted in both MCF7s or PC9s. And so we then concluded that yes, this region is probably driving SOX2 expression in those patient tumors that we had been looking at earlier. And we concluded that this enhancer cluster is required for SOX2 overexpression in breast and lung cancer cells. But we wondered, is this region of the genome required in another context? So why would we have this region of our genome that just that becomes active in cancer? That seems evolutionarily like a bad idea. And so we wanted to know, is this region important in another context? And to do that, we really needed to use an animal model. So we had to see whether or not these regions were conserved. And so if we go and look at conservation at the 124 enhancer or the 134 enhancer, we see that each of them has this deeply conserved core that's even conserved to birds and amphibians. But importantly, they are each conserved in the mouse, which means that we can now delete these enhancers in the context of the mouse genome and ask, is there any phenotype of normal development or adult function that we can see in this enhancer deletion? And so we did that. Again, we used CRISPR to make a deletion of these regions. And the first thing we looked at was just viability at weaning. And so what we found was that, in fact, we found no mice alive at weaning that had the enhancer deleted on both alleles, whereas we found a number of mice that had the enhancer on one allele still alive at weaning. And this was from a total of 46 mice. So we concluded that deletion of this enhancer is in fact lethal. And we decided to look at E18.5, which is just the day before the mice are born. So we looked and we were actually interested in looking at airway and lung development because of our understanding of SOX2's role in that context. And what we found was this very striking phenotype whereby the enhancer deleted homozygotes don't separate out the esophagus and the trachea into two tubes. So this happens normally during development. By E18.5, this has already happened. And in wild-type embryos, you can see that they have a separate trachea and esophagus. And the same was true for the monoallelic enhancer deletion. But in the biallelic enhancer deletion, we saw that all of the mice that we looked at just had this single one tube and failed to separate the trachea and esophagus. And they also don't express SOX2 in this tissue, which they normally do express SOX2 in the cells that line these tubes. And so this phenotype, this would actually cause death shortly after birth due to an inability to both breathe and eat. And so that's likely what's happening and why we don't see any mice alive at weaning. Okay, so in summary, I've shown you that for SOX2, there's a number of different enhancers that are regulating the expression of this gene in different contexts. Close to the gene are enhancers involved in neural lineage commitment and neural lineage phenotypes. Further downstream is this embryonic enhancer that's required during early development. And then close to that, we found this trachea esophagus developmental enhancer that gets co-opted and reactivated in breast and lung cancer. And in the field, this has widely been termed enhancer hijacking. Another group also identified that SR2 within this cluster of neural lineage enhancers is implicated in driving SOX2 expression in glioblastoma. And so just in summary, my lab is working towards a more sequence level understanding of gene regulatory elements and their role in disease. And we're using these deletion approaches as well as machine learning to try to understand the sequence at a deeper level. And what we've learned so far is that genes are often regulated by an ensemble of enhancers, so not just one enhancer, with tissue-specific activity. Enhancers are highly context and tissue-specific, and so understanding this regulatory relationship is very complex and requires looking at different tissue contexts. And the developmental enhancers for SOX2 are reactivated in multiple cancers with different implications for patient treatment based on the underlying active regulatory network that is actually driving those enhancers. So remember, in the breast lung cancer, there was one enhancer that seems to dominate, or two enhancers that dominate, whereas in glioblastoma, which is a brain cancer, there was a different one that seemed to be involved. And just to share my acknowledgements from my team that has, worked on these projects together with the main first authors who I highlighted, and our funding, as well as our collaborators on the breast and lung cancer projects. And thank you, everybody, for your attention.
Awesome. Thank you, Dr. Mitchell, for that talk. That was a lot of really cool work. We'll open the floor now to questions. Again, feel free to type in the chat or raise your hand and we can unmute you. Okay, I can start again. I have lots of questions after this talk. One of the ones that I was thinking of is with the enhancers, is it just open chromatin that's regulating when they're being used, or like how far back can you trace that? Because something must be opening the chromatin. So like how far down can you go in the mechanism?
Yeah, so it's not just open and accessible chromatin, which is why we do have to do the functional assays. So for example, I told you that those gene proximal enhancers in embryonic stem cells, they already have transcription factors bound. They are accessible, but when we delete them, we don't see any effect on SOX2 expression. So, it's more than just accessibility. And from a lot of other work, some of which is published, some of which is unpublished, what we think is happening is that you really need to recruit a large complex of transcription factors. So usually on the order of more than 10 different transcription factors have to be recruited to one of these regions. And that then is what's actually creating an active enhancer, probably through mass action of recruitment of co-activators. So if you think about recruiting this, again, large number of co-activators, like the mediator complex is 30 different proteins, if you can recruit them by using, an ensemble of different proteins, you're going to recruit a much better co-activating complex than if you're just trying to recruit them with one factor, right? So that's kind of a little bit how we think things are working.
Right.
And we can see evidence of that in the sequence. So enhancer sequences generally are on the order of 10 conserved transcription factor motifs for the cell type context that we're looking at.
Okay, that's cool. And then I guess in different types of development, or I guess different contexts when you're using different enhancers, is it mostly just more expression of these proteins leads to more of these transcription factors, or are there protein-protein interactions or PTMs that are regulating how many transcription factors are present?
Yeah, probably both, so a lot of transcription factors are regulated by PTMs. We don't always really know what each of the PTMs is doing or have an idea of what the full scope of them is. So my lab found this other regulation of KLF4 previously where it can be caused to exit the nucleus when it gets phosphorylated on a particular residue. And so, there's this kind of complex regulation of transcription factors. Sometimes we don't have as much information as we'd like to. The kind of classic thing is the signal responsive ones that are responding to some kind of extracellular signal by acquiring some kind of PTM that allows them to then enter into the nucleus, right? So that's the kind of more classic signal responsive transcription factor, which is definitely playing a role. And again, some of them we know a lot about, and some of them we probably don't. So we can't always see that by just looking at expression, But by looking at things like footprints in ATAC-seq data, we can see motifs for transcription factors that might not be showing any different expression in a signal responsive way, but they're probably responding, by some kind of PTM. So we can often see evidence of that by looking at transcription factor footprints on accessible chromatin, where we wouldn't see anything interesting if all we looked at was the RNA-seq, expression levels for the transcription factor. So
Right.
yeah,
That's cool.
long-winded answer.
No, that's good. Thank you. Any other questions from anybody else in the audience?
Hi, I have one question.
Sure.
So it's a great talk, Dr. Michelle. I was wondering, like, how far is the enhancer do we consider for, like, suppose if we have some coding or non-coding gene.
Are you
how
asking
far-
how far away can an enhancer be from a gene it regulates?
Yeah.
So there's a very good classic example of one that is a megabase away, so 1,000,000 base pairs away, and that is the sonic hedgehog enhancer, which is actually in the intron of another gene, and it regulates limb development. And so if you delete that enhancer, you get this truncated limb phenotype in mice. And in humans, it's linked to polydactyly, to digit number. So there's polymorphisms known in that region in humans that are linked to the number of digits. So that's one of the best well-characterized very distal enhancers that is 1,000,000 bases away. But there are other examples of ones that are more than 1,000,000, like 2,000,000 bases away. And there are some examples of ones that seem to work from other chromosomes in mammals at least. And the one that's the best characterized in that respect is the olfactory receptor enhancers. So olfactory receptors for smelling, right? In each olfactory cell, you're only supposed to express one olfactory gene. And what's thought that happens is, there's a collection of olfactory receptor enhancers, but that they will only turn on one olfactory receptor gene in one cell. But it can't always be the same gene. Otherwise, we wouldn't smell a complex mixture of smells. So in different cells, it has to turn on different genes. And so those enhancers do seem to can function from different chromosomes. But that is the most sort of concrete example of something that's not normal in mammalian genomes, quite common in Drosophila, but not in mammals. Thank you for your question.
Yeah, thank you. Thank you.
Okay, seeing no other questions and seeing that we're at time, I want to thank both our speakers again. These were really great talks, and thank you all for coming. And I hope you have a good rest of your day. See you all later.
Bye. Thanks, everyone.